Although HIV is currently a treatable disease, it is still incurable. Treatment, even if possible, is heavy on patients and has great cost impacts. The disease affects over 34 million people worldwide. Each year, another 2.5 million are infected with the virus.
We have condoms and we rely on them enormously as a public health tool. We know at an individual level, if people use condoms consistently and correctly, they really offer the best protection, not just against HIV, but against all the other sexually transmitted infections. But if they were good enough to reduce HIV or get rid of it or control it, then we would have control of HIV, whereas we don't. So they're not enough on their own to control the epidemic. We need something more so that people can control the risk of their infection.
Scientists around the globe work on different ways to reduce this risk. Those involved in the CHAARM project wish to do so by looking for alternative prevention methods. CHAARM is a consortium of 32 partners from countries in Europe, South Africa, and the United States.
The main goal of the CHAARM project is to develop a combination microbicide and at the same time to try and develop some new compounds. We've taken two existing drugs and put them together in a novel combination, and we're testing those both in animal models and in a human phase 1 safety trial. So that will be the main outcome. But at the same time, we've developed 2 or 3 new compounds that show quite a lot of promise and that will be tested in animal models, but they won't get as far as human studies.
But what are microbicides?
Microbicides are formulations of drugs that are applied topically to prevent the virus infecting the first cells if anyone's exposed to HIV. And they're designed to be user-friendly, so they can be used every time somebody's having intercourse.
The microbicide that we're developing is a combination of 2 drugs rather than the single drug that was used in the previous trial.
The combination of dapivirine and darunavir works well because the 2 drugs, we believe, by modelling them in the laboratory, work better than either drug alone. Essentially, it provides you 2 chemical barriers instead of 1, and that hopefully will give us much better levels of protection in humans.
At the CHAARM project, scientists make use of tissue models for many of their laboratory tests. These models are an alternative for clinical trials, which are generally very costly.
The advantage that a tissue model gives us is that it has all the cells in the right context that would be the same case as a human. And we can model whether the drug is safe and whether it blocks the virus from infecting the first target cells that it would be exposed to in a natural environment. So in effect, this is as close as we can get in the laboratory to modelling what would happen in real life.
Before the end of the project, a phase 1 safety human trial will be performed.
We have made both a darunavir only gel and a darunavir plus a dapivirine gel, and we're going to try that out in some healthy women, and we're going to check that it's safe and they don't have any reactions to using that gel. We can actually take the secretions in the women over that time period and check the levels of the drugs that we're administering, and also whether they would block HIV in the test tube. So we can do some nice tests to see if the secretions are actually neutralizing the HIV virus.
I hope that we'll be able to take some of these products further. So the next stage would be a larger human trial, perhaps a phase 2 trial. So we'll need to explore ways of getting funding and support to take them that way. CHAARM's been a very good example of people working together and collaborating, and I think aside from the products that I mentioned earlier, I think one of the lasting legacies will be continued collaboration between partners.
The development of a combination microbicide in the form of a gel or a slow-release vaginal ring would provide an easier and more discreet way of protection against HIV infection.
One extra thing that I know that the very big study that we did was just how much more pleasurable it made sex, because of course they give a bit of lubrication. So I can't think of a better thing for prevention than having something that people want to use.